Hello, my name is Richard Mason, and today I'll be presenting on my poster, Borderline Personality Disorder, Childhood Trauma and Epigenetic Changes, a brief review of NR3C1 gene methylation. So, borderline personality is a complex psychiatric disorder involving intense emotional instability and mood swings, rejection sensitivity, impulsivity, and self harm and suicidality. BPD is prevalent in about 4% of the general population and is underpinned by a complex interaction of biological, genetic, and environmental factors. It is more prevalent in women than in men. Childhood trauma is one of the biggest predictors for BPD development and has been linked to a range of social and cognitive impairments through epigenetic mechanisms. Dysregulation of the hypothalamic pituitary adrenal axis, or HPA axis, has been linked to traumatic experiences. The HPA axis manages the human cortisol response. Cortisol is a corticosteroid released from the adrenal cortex that binds to glucocorticoid receptors. Impairment of these glucocorticoid receptors has been identified in HPA dysregulation in BPD. The NR3C1 gene, responsible for GR protein transcription, has become a focus of epigenetic research. Methylation of this gene has been linked to BPD symptoms by inhibiting GR production. Recently, NR3C1 methylation, particularly in the promoter region Exon 1F, has been investigated in a relationship between CT and BPD symptoms. Due to the exploratory nature of the neuroscientific research in the BPD field, methodological inconsistencies have appeared in DNA research. These have culminated in varying demographics, designs, and statistical analyses being employed by researchers. Standardization is beginning to unravel the molecular mechanisms underlying the pathology, though. Up until 2011, NR3C1 research had only compared methylation with BPD diagnosis generally or particular symptoms, but hadn't really tried to connect the dots between childhood trauma and BPD. This review will contrast the approaches and results of three papers that applied the same DNA methylation analysis procedures to investigate how methylation of exon 1F of the NR3C1 plays an epigenetic role in connecting CT and BPD. This will then be placed in a wider context of BPD research overall. The details of the three studies can be found in the reference section, but the first study was a 2011 study by Perroud and colleagues, Study 2, a 2014 article by Martin Blanco et al., and finally a recent 2021 study by Flasbeck and Brune. Onto the methods. This is a condensed table that I created comparing the methods of the three studies. First thing to note is the use of clinical groups differed between the three studies. The first compared BPD with MDD and included a small MDD and PTSD contrast group. The second had one BPD sample and compared those with CT experiences with those without. The final study compared BPD with healthy controls. A second point to mention is that one study did not include males at all. Although other studies had less than 15% of males, which is representative of the general population, of the general BPD population, this could play an influence. Finally, Study 2 excluded comorbidities due to the different and sometimes oppositional effects different disorders have on HPA functioning. The other studies had over 50-60% comorbidity rate, which is standard for BPD population, but it does introduce some interesting factors. Each study used different diagnostic criteria, though this is expected with how the DSM and other diagnostic tools have been updated over time, along with our understanding of BPD. One thing all studies did identically, though, was the application of the Childhood Trauma Questionnaire, which measures childhood trauma in 5 subscales: physical and emotional neglect and abuse, and sexual abuse. This comes in handy because at least we know that they're measuring the same childhood trauma patterns in each study. On to the procedures, so each study did employ the same steps for DNA methylation analysis. Sample collection, bisulfite treatment, PCR amplification, and pyrosequencing. This process differentiated methylated cytosine nucleotides located at the CPG sites by transforming and then measuring unmethylated cytosines in the DNA strain. The first 2 studies targeted the first 8 CPG sites along exon 1F, located between 2 nerve growth factor induced sites that had been highlighted in a previous BPD study. And study 3 analysed 42 CPG sites using next-gen sequencing. And a couple of these studies actually included other measures to test the effectiveness of these techniques that were used, and it has been shown to be as reliable as other DNA techniques. The source of DNA sample collection differed between the studies, though. Studies 1 and 2 sourced DNA from leukocytes obtained from peripheral blood samples, in which, provide similar methylation data to direct brain tissue samples when looking at NR3C1, so this seems pretty sound. And study 3 used mouthwash to collect an oral sample, but a justification for this was not offered in the article. Results. So, study 1 reported that for the whole sample, child sex abuse and the number of and severity of traumatic experiences during childhood were correlated with NR3C1 methylation. Increased methylation was associated with repetition of CT generally and with penetrative sexual abuse specifically. Study 2 found that BPD severity overall, as well as self-injury and history of hospitalization, were associated with increased NR3C1 methylation in BPD patients. A linear regression analysis determined that physical abuse, previous hospitalization, and the Dieben score, which is the BPD severity measure used in this study, taken together predicted 16% of the variance in NR3C1 methylation. Study 3 recorded decreased methylation, however, in the BPD group compared with health controls. The difference between the groups was only 0.08%, and there was no connection with CT. Individual CPG sites were analysed. Study 1 reported a greater number of traumatic incidences during childhood was positively correlated with increased methylation in CPGs 2 to 8. Only Study 2, though, analysed the methylation of individual CPG sites across all variables, as shown in the table above. Emotional neglect, physical abuse, and emotional abuse were associated with methylation at specific sites. Previous hospitalization and self-injury were correlated with specific CPGs, in addition to overall methylation. The Dieben was also additionally significant in the overall CPG analysis, and the third study did not run any CPG analyses like individual sites. Overall, the results demonstrated a support for increased methylation being a link between childhood trauma and BPD symptom severity. Firstly, Study 1 identified that the number and severity of incidences during childhood predicted increased methylation, with child sex abuse being the most significant adversity. Study 2 provided support for these findings and was the first to run CT, BPD, severity, and methylation in an analysis together, revealing a significant relationship between the three factors. Calculating methylation in individual CPG sites can be critical to identify some of those smaller effects and to avoid masking. Study 1 revealed this relationship that's discussed earlier, that the number of CT experiences increased methylation at 7 sites. Study 2 reported methylation at 4 CPG sites. It was connected with CT subscales, and 5 are connected with BPD severity scores. And 2 of the sites are actually correlated with both. As mentioned, Study 3 did not analyse any CPG sites and also lacked sufficient power to make the results significant. So I think it was about 0.757, so just probably under the threshold of what we'd be looking for. One thing to note is that despite the CPG sites were different for each studies and different from other studies, this is sort of contributing to the argument that an individual CPG site does not have a specific type of childhood trauma or symptom connected to it. However, any symptom or trauma, and it's just the incidence and the severity of that trauma that does methylate a site, and it's not tied to any particular CPG sites. Still, the results of studies 1 and 2 were small, and there are likely more variables moderating the relationship between CT and BPD. Potential competing biological explanations include other epigenetic processes, such as microRNA, which has been identified in one study for its regulation of the NR3C1 transcription process. Other NR3C1 exons, so 1B and 1C, are the most common forms in the brain, despite 1F's abundance in the hippocampus. And the latter 1C has been identified with BPD symptoms in bulimic patients. Other HPA excess genes that might moderate the HPA dysregulation, such as FKBP5, which Study 3 also looked at, and other biological systems like the serotonergic system that is but plays a role in BPD generally. Despite each being connected to BPD development, though, none are yet to provide a competing explanation for the biological link between CT and BPD. NR3C1 exon 1F offers the strongest known evidence for epigenetic interactions with childhood adversity and BPD symptoms. This is a crucial finding as this backs up a lot of other neuroimaging and systems level studies that have reported this relationship and its connection with HPA dysregulation. So it does certainly seem that methylation of the NR3C1 inhibits GR receptor coding. Though more research is needed to quantify this and to identify other biological factors involved. But thank you very much for listening to my presentation.